So, a very good evening to everybody present. Today, we are going to do questions. Just a minute. Yes. So, today we are going to do questions for week 7. Okay. So, we'll be doing questions of week 7. So, in week 7, the topic that is started is the Drosophila axis development. How axis development in Drosophila takes place. So two parts have been covered in week seven's lecture, and then subsequent more two parts will be covered in next week's online class. So I hope you all have gone through the lectures and you have gone through the videos that were provided to you, because Drosophila axis development is a very important topic, and as much as important it is also a very lengthy topic. So when I say it's a lengthy topic, it means that the concept development and question solving cannot be done in the same 2 hours class. So I assume that you have gone through the lectures and you have a basic understanding of how the axis development, the dorsal ventral axis and the anterior posterior axis takes place. So whenever we, this is like one of the most interesting and one of the most important part of developmental biology, also a part which is often asked in your competitive examinations, for especially your CSIR examinations. So if I talk about the two most hot topics that are being asked in CSIR, the first is your this Drosophil axis development, and second is little bit in B section, your stem cells and properties of stem cells and questions related to stem cells and IPSCs is asked. So this is an important topic, and why I say it's an important, because understanding the theory is one thing and application of the theory is another thing. So you would have questions where you have to apply the knowledge that you have gained through the lectures or like wherever from wherever you are reading it, and then you have to apply it through various examples, through various experimental ways, that if something is deleted, what will happen, what would be the phenotype, what would be the formation, how would be the formation, so like this. Okay, so now we'll start with simple questions and then we'll try to solve one or two experimental based questions. Fine. So our first question for the day is, hmm, which of the following which of the following will not result in anteriorization of the Drosophila embryo? Your options are: option A, expression of dorsal at the interior; B, transcription and translation of the hunchback at the posterior; C, translation of nanos at the anterior; and D, expression of bicoid at the posterior. Okay, you have 2 minutes to think and then we will switch the screen. It's an easy question. 1 minute more. Okay. I'm switching the screen now. Hmm. Let us see what you have answered. Hmm. Hmm. So Ajay has answered C and D. Sorry, A and C, and just Reena has answered C and D. And okay, let's see. The question is, which of the following will not result in anterization of the Drosophila embryo? And the accepted answers are expression of dorsal at the anterior and translation of nanos at the anterior, which is A and C. Okay, A, is it A and C now? Yeah, expression. Yeah, it's A and C. So A and C are the correct answers. Now we'll try to analyze each and every statement. First of all, let me mark the correct answers. So the correct one is hmm, sorry, yeah. The correct one is A and C. Okay. Now let us try to see. The question says that which of the following will not result in anteriorization of the Drosophila embryo? So Drosophila has an anterior axis and a posterior axis. Okay, there is an anterior axis and there is a posterior axis. So you should know the basic uh, morphogens or the basic proteins or mRNAs depending on which stage we are talking about, which are specifically involved for the anteriorization and which are specifically involved for the posterior side for formation. So, bicoid is responsible for what? Anterior or posterior? Tell me. Bicoid is for anterior or posterior? Anterior. Good. So, basic, you should know. Bicoid is for the anterior axis. Then nanos is for anterior axis or posterior axis? Posterior axis. So nanos is involved in posterior axis. And hunchback is for anterior axis or for posterior axis. Hunchback, anyone else? Just Reena, do you know? Hunchback, anterior or posterior? Anybody knows? So hunchback can act as both, but mostly in the starting, hunchback will help in the anterior axis formation because it is activated by bicoid. And then caudal, caudal, anterior or posterior? Anybody knows? Caudal. It is involved in anterior axis formation or posterior axis formation? Tell me. Anybody else? Caudal is involved in posterior axis. Okay, so you should always remember that nanos inhibits your hunchback and bicoid it inhibits caudal. Okay, remember that nanos it inhibits your hunchback and bicoid it inhibits your caudal. Okay, now detail we won't go right now. We'll now try to solve the options which are given in the questions. First option is expression of dorsal at the anterior. Now, dorsal is a protein which is involved in dorsal to ventral polarity formation. If you know, dorsal to ventral polarity formation, dorsal is involved. But important point to note is that dorsal is involved for ventralization. Okay, so dorsal is involved in establishing the ventral polarity and not the dorsal polarity. So, whenever we talk about the names of the various proteins which are involved in your developmental biology, they basically tell the mutated uh, phenotype. So, if dorsal is not expressed in the ventral side, it leads to dorsal side formation. Therefore, it is known as dorsal. So here the question is that which will not cause the anteriorization of the Drosophila embryo. मतलब anterior formation will not take place in which of the cases? First is when dorsal is expressed in the anterior. Okay. Our options are expression of dorsal at the interior and translation of nanos at the interior. So when dorsal is expressed in the anterior side, then in the anterior side also your ventralization would take place. Could you understand what I'm trying to say? See, just a minute. See, we have dorsal to ventral axis formation, in which one of the key players is dorsal. Dorsal is responsible for ventral axis formation or ventralization. That means when dorsal is expressed, ventralization occurs. So if dorsal is expressed in the anterior side, the nature of dorsal is to promote ventral axis formation. So if it is uh, expressed in the anterior side, it will again start ventralization, ventralization at the anterior end. Okay, so dorsal will start ventralization at the anterior end. So anterior axis would not be formed. So in this way, option A is correct. Understood? Because dorsal is a protein which is responsible for ventralization of the Drosophila embryo. That means dorsal protein helps in ventral axis formation. So see, always remember whenever we're talking in developmental biology, it doesn't means that because dorsal is responsible for ventralization, so dorsal will work only when it is present in the ventral axis. No, dorsal can work anywhere. The phenotype that it will give is a ventral axis formation. So if dorsal is expressed in the anterior end, it will form ventralization only. It will not switch to forming in the anterior axis. This is very important thing that you should understand whenever you're trying to solve questions of developmental biology, that any protein, irrespective of the place where it is present, whether it is present in the anterior side, whether it is present in the posterior side, dorsal side, ventral side proteins, it will show its property irrespective of the place. Okay. So dorsal protein is responsible for ventralization. It will ventralize even if it is present at the anterior end. So A is the correct option. Then transcription and translation of the hunchback at the posterior. So hunchback is responsible for the anteriorization. That means for the anterior axis formation. So if hunchback is also present at the posterior, what would be the phenotype? What it can lead to? Can anybody tell me? If hunchback is also present at the posterior end, what can be the possible phenotype if it is present at the posterior end? Anybody? So anterior hunchback is expressed in the anterior end. If it is present at the posterior end, that means posterior axis formation is hindered. Posterior axis formation is hindered, not the anterior axis. Anterior axis is well preserved. So this phenomena or this will not lead to any problem in anterior axis formation. There can be a problem in posterior axis formation, but not in the anterior one. Then Translation of nanos at the anterior. So nanos also specify the posterior axis. So if nanos are present in the anterior axis, then what will happen? Ventralization of the anterior axis will take place. Okay. Then expression of bicoid at the posterior. So if bicoid is present at the posterior axis, then posterior axis would be anteralized. That means anterior axis formation will take place at the posterior end if bicoid is expressed at the posterior end. Okay, see, that's what I was telling you in the starting, that these questions can be very confusing if your concepts are not properly built and if you're not practicing the questions. So as many questions as you will practice, then only you will get to know. So I hope this question is clear to everybody. You need to understand very important thing, that bicoid and nanos have just opposing role. Bicoid helps in anterior axis and nanos help in ventral axis. Similarly, hunchback has a major role in anterior axis formation, whereas caudal has a major role in ventral axis formation. Fine. Yes, Ajay, you can ask your question. You can type it in the message box. We'll see, bicoid is everywhere. After I understand what you're trying to say, but you need to understand, you need to accept this point also, that initially when there is no fertilization has taken place, the bicoid mRNA is present in the anterior axis. Okay. As soon as the fertilization occurs, the bicoid mRNA is converted into bicoid protein. Now, this bicoid protein forms a gradient, which is known as a morphogen. Like it acts as a morphogen, it forms a gradient. So, the place where bicoid protein is present in the maximum amount, that forms your anterior side, that is the head side. And then, as soon as the bicoid levels decreases, in that line, it forms thorax. So, this is how it happens. What I'm trying for everybody to understand is that it's fine that bicoid helps, bicoid gradient defines the segments which are formed and the axis which is formed. But overall, you should understand for solving the questions per se, that bicoid helps in anterior axis formation and nanos helps in ventral axis formation. The concept that bicoid after fertilization becomes bicoid protein and then its gradient decides whether it will be head and thorax, that will come later. Okay. Now, we come to the next question. Hmm. If bicoid is added to the middle of the bicoid deficient embryo, which of the following is or are the expected embryonic pattern? Your A stand for acron, AB for abdomen, H for head, T for thorax, and TE for telson. Your options are AHTABTE, TEABTE, telson thorax head, thorax abdomen telson, abdomen head, thorax abdomen, thorax head acron, and embryo will not be viable. So there can be more than one correct answers. I'll give you 3 minutes to solve this question. Okay. Try to solve the question. Anyways, we are going to take each and every question in very detail. Okay. Doesn't matter how many questions we are able to solve. Our goal is to at least understand the concept of the questions that we are dealing with. So your time starts now. It's 6:20. I'll switch the screen in 6:23. Okay. 1 minute more. See all the options very carefully. Try to make a diagram, uh, what happens in the wild type condition, and then try to compare it with the mutant one. Okay. So if you know how in a wild type how it works, then it would be easier for you to solve this question. Hmm. 1 minute more. Your time is over. Okay, okay. Okay, fine. I'm going to switch the screen now. Hmm. Okay, let me see what are the correct answer. Okay, fine. Uh, suppose bicoid. Okay, Ajay has answered. Suppose bicoid is nil, can only post axis formation E and C. Better if C will be telson, abdomen, thorax, head, thorax, abdomen, telson. Okay, fine. So anybody else who wanted to give a try? Anshika, Jasrina, Shubhamoy, Fatimuz. I'm sorry if I'm spelling your pronouncing your name incorrectly. Anybody who thinks or anything you want to say? Ashika, no ma'am. So you don't have you read these topics, Ashika, before? Like these topics, this developmental biology? Okay, so you haven't, this is your first time. So have you gone through the lectures then? So for the people, I would want to say that for the people who haven't read these topics before, along with the lectures, you should also try to read some book. Okay, so the best book for developmental biology is Gilbert. Okay, I'm typing it, Gilbert. So just read this book, go through the topics which are at least there in your syllabus of NPTEL course. You will have a very good idea. So this Gilbert is a very good book. It's a standard book for developmental biology. Take any latest edition, but read, because without reading, relying just on this class or the recorded lectures will not help you to gain the concepts. You have to read it, because not everything can be covered in a 2 hours class. Okay. Anyways, now we'll try to solve this question. So let us see what happens in a normal condition. That means in a normal wild type condition. Okay. So in a normal, which we can say as a wild type condition, what happens? What comes? What is the anterior most part in the normal condition? Anybody? What should I start with? First is in a normal condition. People, okay, you couldn't answer the mutated version. At least if you know the uh, normal way how it happens. First is what? Abdomen, thorax, head, telson, acron. What comes first? I'm starting from the anterior end. First comes acron. So first is your acron. I'll just write A. Then comes your head. Then comes your thorax. Then comes your abdomen. And then comes your telson. Telson is specifies the tail. This is head, thorax, abdomen. Telson is the tail. So this is the normal case. Okay. So a wild type case has this. Now, this head and thorax that has been formed, This portion, the anterior portion, has resulted because of the protein gradient of what? Bicoid. How? Sorry. So, before the zygote has formed, before the fertilization, so bicoid mRNA are present in the anterior realm. This is an anterior, this is your bicoid mRNA. Okay. After the zygote is formed, this mRNA gets translated and it is converted into protein. That is bicoid protein. Now this bicoid protein forms a gradient. Okay. So this bicoid protein forms a gradient like this. There is a gradient. And this gradient decides whether head thorax would be developed or not. So here the gradient is more, here the gradient is like slowly the gradient is decreasing. That means the concentration of bicoid slowly decreases in this way. So where the concentration of bicoid protein is more, there your head forms and then forms your thorax. And then in the posterior side is your abdomen and telson tail. Fine. Now, the question says that see, the question says that the embryo is deficient of bicoid. Now, if an embryo is deficient of bicoid, that means there is no gradient formation that has been taken place. And if there is, yeah, because there is no bicoid, so both the regions will function or will work as ventral side and what will happen, see, if you're getting confused, so in the posterior end is your telson, because it does not has bicoid. Here also there is telson. That means there are two tails. Okay, telson is there and abdomen is also there, because abdomen is not dependent upon the bicoid, it is dependent upon the posterior signals. Now they are saying that this is the mutant embryo which does not has bicoid gene, it is bicoid negative. Now in the middle, bicoid has been added, bicoid has been added in the middle. So what will happen? What will happen? The middle part will have the most concentration or the maximum concentration of bicoid. So this will form head. Okay. Now, if see in normal case, what happens that this is your bike from here, it the concentration like goes on. So here this is your head, here is your head, and then the concentration decreases, or so you have thorax and all. Now here what can happen, this bicoid can diffuse this side also and the bicoid can diffuse this side also. So what will happen, you will have thorax here and you will have a thorax here. So your final phenotype would be telson, thorax, head, thorax, abdomen and telson. This would be the final phenotype. Let us see if this is the answer. Huh. Yeah. And because because your anterior and posterior for anterior formation is not proper, therefore obviously the embryo will die. It is not a viable embryo. That is fine. But you need to understand this part that I tried to explain you. So the part, that's what I was telling you, that why we say that bicoid is responsible for anterior axis formation, because the portion which is receiving the maximum amount of bicoid protein, that particular protein portion will form the head. And then there is a gradient form, and since your bicoid is inserted into the middle, therefore gradient formation will occur in both the directions. If in the normal case, what happens? Gradient formation occurs in this direction because it is going like this. But here, since it is your bicoid is added into the middle of the embryo, so in both the directions your gradient formation will take place, and in both the directions your thorax would be formed. Okay, I hope it is clear to you. Is it clear, Jastrina? Ashika, is it clear to you? Okay, yes, Ajay, you can raise your query. You can write it in the chat box, whatever query you have. Fine. So even see, I am not expecting you guys to understand it in the first go at the very first place. Okay, I understand that this is a not a difficult topic, but a conceptual topic. Fine. So it will take time for you to understand, but eventually you will get to know. Fine. See, abdomen will not be present on both the sides. You have to go to the very basics. I won't be explaining into very much detail. But as your anterior side is determined by your bicoid and hunchback and other things, your posterior portion, which specifies your abdomen, is responsible because of some other proteins such as nanos and caudal. So, because the expression of nanos is maximum at the posterior end, therefore abdomen is formed in the posterior end only. Because for abdomen formation, for head formation, we need the specific expression of its specific proteins, that is nanos and this bicoid. That is why. Okay. Now I'll give you a condition. Okay. What will happen if in a bicoid deficient embryo, bicoid is added at the uh, I will give you two questions. First is anterior end, simple. And this is the first question. Second question is what will happen when in a wild type embryo bicoid is added to the posterior end. Okay. So these are the two questions that you have to solve. First question is in a bicoid deficient embryo, if you add bicoid at the anterior end, then what would be the phenotype? Second question is in a wild type embryo, if you add bicoid at the posterior end as well, then what would be the phenotype? Okay, I am giving you 5 minutes to solve this question. Let's see whether you are able to understand the concept or not. This question is, see, Ajay, if you know the answer, please at least wait for 5 minutes so that other students can also uh, think and answer, because I don't want anybody's answer to get influenced by the answers that has already been written on the message box. So it's my humble request to everybody, to please answer at least after 3 minutes, okay? So that everybody can keep the same pace. So, this is the question. I'll give you 5 minutes. Think and answer. And I want Jasrina, Ashika, Fatimuz, Shubhamoy um, to also to try. Thank you, Ajay. Hmm. Okay, chalo. Your time starts now. Think. We have already dealt the phenotype of a wild type embryo, and we already know the little bit about the deficient embryo also. I want both the answers at the same time. That's why I've given you 5 minutes, okay? 5 minutes is not just for one answer, 5 minutes is for both the answers. Think and try to solve the question. Okay, 2 minutes more. One more minute. Okay, I'm switching the screen now. Let's see. Hmm. Okay. Okay. So Ajay has answered A would be normal development. Okay. Okay, I have to see what are the options. Okay. A is normal development and B is two heads form anteriorly and posteriorly. So very good. Ajay has answered it absolutely right. And Ashika has also answered something. Let me see what she has answered. So I can see that just Reena has already left the meeting. Maybe she got scared that I will ask her. Okay. Anyways, so what I'm trying to say is that don't get afraid or scared that I'm asking questions. Okay. I'm not giving you any marks. Even I'm a PhD student, I'm not even a teacher. I'm also a student. I'm doing PhD. So we're just trying to learn from each other. Nobody is perfect here. But if you try to solve, it will increase or improve your thinking ability only. Okay, it is helpful for you. You are solving, you are thinking, you are trying to think conceptually and intellectually. It will help you. It's not going to help me. Fine. So even if you're answering it wrong, it doesn't matter. I'm not going to judge you. Okay, I'm also a student, but at least you're trying, and that is what really matters a lot. Fine. So first of all, I want to appreciate Ashika that she tried to answer the question. Now let us see what she has answered. I referred with the basics. I tried to answer the following question as now of what is understood is bicoid is responsible for anterior development if present at the dorsal okay. So see, you have already answered the second one. See, you have written that bicoid is responsible for anterior development if present at the dorsal, and that's the answer of the question, that if bicoid is present at the posterior side, then what will happen? If present at the posterior end, just remember it is anterior, posterior, and dorsal ventral. Okay, so if bicoid is present in the posterior side, then in the posterior side also it will form head. So it will be two heads. See, you have already answered it. So that's what I'm saying. That try to understand the concept. You can answer it. It's not very rocket science that you cannot understand. Fine. So now let us try to solve this question. So the first one was bicoid deficient embryo. Bicoid is added at the anterior end. So when bicoid is added at the anterior end in a bicoid deficient embryo, it will resume its normal phenotype. As simple as that. See, this is your embryo. Fine. It is bicoid deficient. Now, when you are adding bicoid at the anterior end, then what will happen? Acron, head, thorax, abdomen, and telson. Because since it is added at the anterior end, so in this way, your gradient will go. And when it will go in this way, your first head would be formed, then thorax would be formed, then abdomen, then telson. So when bicoid is added at the anterior side to a bicoid deficient embryo, it would resume its normal development or normal phenotype. Then next question was, if a wild type embryo, in a wild type embryo, if bicoid is added to the posterior end also. So if bicoid is added in the posterior end also, that means here head and thorax forms. Here also, depending on the concentration, here also head and thorax would be formed, and in between there would be abdomen, and there would be acron, and there would be acron. No telson formation takes place. That means this embryo, sorry, this embryo has a phenotype with two heads. Okay, so just a basic, even if you're getting a basic understanding, it's fine for me. Now we'll come to the next question. Oh, how has this come here? Okay, just a minute. Are okay, I think this is some technical problem. Anyways, so the next question is, which of the following pairs of genes are directly regulated by bicoid? Krupal and caudal. Second option is caudal and krupal. Third option is nanos and hunchback. Fourth option is caudal and ribs. And your last option is hunchback and caudal. Okay, it's 6:47. I'll give you 2 minutes. So I'll switch the screen at 6:49. Okay, try to answer the question. 1 more minute left. Okay. Okay, so I'm switching the screen now. Okay, fine. So, Ajay, okay, nanos and hunchback, and Ajay has answered hunchback and caudal. Okay, so I think, hmm, which of the following pairs of genes are directly regulated by bicoid? So, I have told or not, but I'll just tell you again that bicoid. Okay, first of all, we'll see the answer. The answer is hunchback and caudal, which is this one. Sorry. Yeah, this one. Okay. Hunchback and caudal are the right answers. So, how hunchback and caudal are the right answers? You see that bicoid promotes the expression of hunchback, and bicoid inhibits the expression of caudal in the anterior region. Okay, so we have to see that which of the following pair of genes are directly related or regulated by bicoid. Now, this regulation can be either a positive regulation or this regulation can be a negative regulation. So, hunchback bicoid activates the transcription of hunchback in the anterior region of the embryo. Okay, so in the anterior region of the embryo, bicoid, it activates the transcription of hunchback. Whereas in the anterior region of the embryo, bicoid represses the translation of caudal protein. And how bicoid? So bicoid, it represses the transcription of translation of the caudal mRNA by binding to its 3' UTR. So you have to understand that caudal mRNA cannot be translated in the anterior region because of the bicoid protein. So therefore, caudal protein, it is confined to the posterior region and it allows the posterior development. So the correct answer is hunchback and caudal. Okay, so Ashika, you have answered one very correctly, that is hunchback. So according to your understanding, I think you have tried to answer it nicely. So you just need to understand that caudal mRNA, if it tries to enter the anterior region, it cannot translate itself because of the bicoid protein. So bicoid does not allow the translation of caudal mRNA by binding to the 3' region of its caudal mRNA UTR. Okay. Now we come to the next question. Uh, okay, fine. This is the question. The zygotic genes which are activated at the posterior end are posterior. Okay, there are two ends, anterior and posterior. We want to see that are activated at the posterior end are bicoid and hunchback, nanos and Oscar, nanos and bicoid, Oscar and bicoid, giant and hunchback, and tailless. It's a very clever question. I'll tell you why. I'll give you 3 minutes to answer this question. It's 6:54 right now. I'll switch at 6:57. Fine. Now start solving the question. 1 minute more. Okay. See what you have answered. Hmm. Okay. यहाँ से. From where you have started, I think. Since Oscar, so Ajay has answered, since Oscar is a protein, so tailless and giant and hunchback bicoid nanos activated before zygote formation. Okay, yes, you are correct. So now comes another important concept that we will try to learn with this question. So with each question, we are trying to learn some of the other concepts. Now, the question says the zygotic genes which are activated at the posterior end. Now, if you do not pay attention to this question, okay, you will think that okay, posterior is written, that means what should be there? Nanos should be there, Oscar should be there. Now you can get confused like this. But you should understand that they have asked about zygotic genes. Why I am stressing over here zygotic genes? There are two categories of genes or two sets of genes. Okay. Can anybody tell me what are those uh, genes? Hmm? Can anybody tell me? Yes, before and after zygote formation. So always remember when we try to study about the various genes which are, sorry, I don't know why this is coming. Yeah, various genes. So the genes can be classified into two major groups. First one are the maternal genes, and the second ones are the zygotic genes. Now, maternal genes are the genes that are that regulate the early development. Fine. And zygotic genes are the genes which are expressed after the zygote formation, like just after the zygote formation. So they regulate the initial events. And after the zygote formation, the later events are in under the regulation of zygotic genes. So bicoid is a maternal gene. Oscar is a maternal gene. Fine. So at the time of when maternal genes are only expressed, at that time, up till the zygote formation, I mean the after the zygote is completely formed, your zygotic genes are not expressed. They are expressed afterwards. That means they are expressed later. So I don't know why it is coming like this. Anyways, I'll do it here. Hmm. So these are maternal genes are the genes which are present in the initially before the zygote formation has occurred, and zygotic genes are the genes after the zygote has formed. Then these zygotic genes are expressed. So they are expressed after the fertilization has occurred. Before the fertilization, only maternal genes are expressed. After fertilization, zygotic genes are expressed. So in name suggests maternal genes are the genes which are present in the oocyte, which are present in the egg, and zygotic genes are the genes which are present only after the fertilization has taken place. That means not just they are coming from the maternal side, they are zygotic genes. Now under zygotic genes also you have subcategories. Okay, so if we try to understand zygotic genes, so these topics that I am telling you, try to read these topics in detail afterwards. This is your homework, that you try to read about maternal genes and zygotic genes. What is the difference between maternal genes and zygotic genes? What are the different subcategories of zygotic genes and their examples? So, zygotic genes can be your gap genes, they can be pair rule gene, they can be segment polarity gene, and there is one more. I'm forgetting the name. Can anybody remind me? The last one that gives the function to the segments that has been formed. The last gene, the last category of genes, which provides or which gives the function to the segments that has been formed. Ajay, do you know the last category of the zygotic genes? Anyways, you can just go and check. So what I'm trying to tell over here is that essentially there are two categories of genes which are present. One category of genes are the genes which are formed before the fertilization, and these are the genes which are expressed after the fertilization. So before fertilization, your bicoid and oscar, like these genes are, and nanos, these are the genes which are your maternal genes, and genes which are formed after fertilization in the zygote are the zygotic genes. Now, zygotic genes can also be of various types, such as gap genes, pair rule genes, segment polarity genes, and there is one more category that I'm forgetting right now. You can just check it. So, these are the various types of zygotic genes. Now, this is one thing that I wanted to tell you. Now, let us again move to the question. Our question was that the zygotic genes which are expressed at the posterior end. So they are specifically asking the zygotic genes. That means, uh, I don't know why this software is creating so much problem today. Yeah, that means bicoid cannot be included, Oscar cannot be included, nanos cannot be included, because these are maternal gene. So, anyways, bicoid is not expressed in the anterior posterior side also. That is also one way to negate that option. So, this cannot be the option. Nanos and Oscar cannot be the option. Nanos and bicoid cannot be the option. Oscar and bicoid cannot be the option. We are left with these two: giant, hunchback, and tailless. Now, giant and hunchback, these are your, and tailless, these are your zygotic genes. So essentially, giant and hunchback are your gap genes. Fine. So these three are zygotic genes. Okay. So one thing is confirmed, that these are not what to say, maternal genes. These are zygotic genes. Now we have to see that whether are these genes really involved in posterior end activation or not. So, I mean, they are really present in the activated posterior or not. So, if you remember in the first in the starting of the lecture, I told you that hunchback. So, always remember this about hunchback, that hunchback is majorly involved in your anterior axis. Hunchback is involved in anterior axis formation, uh, but it is also a little bit hunchback in a very narrow domain. It is also expressed in the posterior region. Okay. So in a very narrow region of posterior side of the embryo, hunchback is also expressed. So hunchback expression is observed, although it is not that much prominent, but still hunchback expression is also observed in the posterior region. Giant. Giant is present in the posterior region, which is responsible for posterior structure formation. And tailless is also a gap gene, and it is mostly responsible for the posterior structure formations like telson. Okay. So tailless gene is responsible for the formation of tail. So if you remember, I told you in the beginning of the lecture that in developmental biology, names are given according to their mutant phenotype. So if tailless gene is responsible for the formation of tail, or sorry, for the yeah, I mean telson. So if tailless gene is absent, then that particular embryo would be tailless. So that is why it is named as tailless. So the genes are named according to their mutant phenotype. So what would be the correct answers? The correct answers would be E and F, because these three genes, these are zygotic genes. More specifically, they are gap genes, and all these genes are present in the posterior axis. See, here they are, it is written which are activated at the posterior end. It is not written which are responsible for the formation of posterior end. Had it been written like this, then we could not have included hunchback there. Because essentially the role of hunchback is anterior axis formation. Although it is present in a very narrow domain in the posterior end only, but it tries to increase the expression of genes or other gap genes in the anterior end. It helps in the anterior axis formation. So from this, we got to know another concept, that in developmental biology, particularly when we are talking about Drosophila axis development, genes are divided into various types. And these various types can be maternal genes and zygotic genes. Maternal genes are the genes which are present before the fertilization in the oocyte itself, and zygotic genes are the genes which are present when the zygote is formed. Some examples of maternal genes are bicoid Oscar nanos, and these zygotic genes are further classified into various subtypes such as gap genes, pair rule genes, segment polarity genes, and etc. Fine. So the question here said that we wanted to specifically answer the gap rule gene, sorry, the zygotic genes. And because we needed to specifically select the zygotic genes, that is why we could not select nanos and Oscar option. I hope this is clear to you. Not segment polarity. Just a minute. I will tell you the name of the last zygotic gene also. So we have gap genes, pair rule genes, segment polarity genes, and another set of genes which give function to the segments. Uh, okay, I think it will take time for me to see. Gap genes, pair rule genes. Okay, anyways, we'll see that later on. Hmm. We'll see that later on. There is another homeotic. Yes, homeotic selector genes. So this is the last category. Gap genes, pair rule genes, segment polarity genes, and the last one, Ashika has answered it very correctly. These are the homeotic selection genes or homeotic selector genes, which gives which assigns function to each of the segments that has been formed through segment polarity genes. So I think in your seventh, I mean eighth week lecture, because your eighth week lecture is already out now. So I believe that in your eighth week lecture, it must be given. I haven't gone through the lectures, eighth week lectures yet, but it should be given, like about the gap genes, segment polarity genes, and all. And if it is not given, I'll try to cover it in the next class so that you have an idea. Okay, because this particular topic is really very important, guys. I'm stressing this again and again because this is a major portion that comes in your CSIR from this developmental biology. So we'll try to cover the different zygotic genes in the next class. If it has already been covered by Sir, it is perfect, and I think it would be covered by Sir, of course. Otherwise, we'll try to have a basic understanding of it. Okay, now we'll come to the next question. Which of the following is or are responsible for ventral fin? Dorsal, ventral, PLK, bicoid, or pipe? Okay, this is your question. 2 minutes to answer this question. After 2 minutes, I will switch the screen. So this is 7:12 right now. I'll switch it 7:14 something. Okay? Now try to answer it. Okay, now 1 minute is left. What's the time? No, no. Time is up. Chalo, I'm switching the screen now. Hmm. So Ajay has answered dorsal and pipe. Okay? And the correct answer is, I think, dorsal and pipe. Yes. Fine. So now we'll try to understand the function of each and every gene. So we have to find out, or we have to tell, that which of the following genes are responsible for ventral fate. That means we want to see, or we want to select the options which are responsible for ventral axis formation, right? Yeah. So I have already told you that dorsal is a protein. I mean, dorsal, yeah, which is responsible for ventralization of the embryo. So, dorsal is expressed in the ventral part of the embryo, and it is responsible for ventralization of the embryo. So, why it is named dorsal then? Because I have told you that in developmental biology, genes are named according to their mutated versions. So if dorsal is not expressed in the ventral axis, then ventral side will also become dorsal. Ventral side will also gain the dorsal polarity. That is why dorsal is named as such, as this. So for ventral fate, dorsal is required. Then is ventral protein required for ventral fate? Is there any protein like ventral? No. Ventral, there is no any specific protein or gene as ventral. Ventral is just the direction in the embryo. Okay, dorsal is also a direction. Dorsal, ventral are the directions. But dorsal is also a protein. I mean, dorsal has, there are two kinds of dorsals. First is the axis, dorsal ventral axis, and second is a dorsal protein. Two different things. But ventral term is only used for specifying the direction. There is no such any protein or gene named ventral. So this is only, it refers, only the direction. Then PLK. What is the meaning of PLK? PLK is polo-like kinase. PLK is polo-like kinase. And what is the function? PLK is basically involved in cell cycle regulation and mitotic process. It is not directly responsible for specifying the ventral fate. Okay, it is not very much involved in axis formation. So PLK, which stands for polo-like kinase, it is responsible for cell cycle regulation and some other mitotic process. So this PLK is involved in cell cycle regulation and other mitotic processes. It is not directly involved in ventral fate. So this is also not involved. Now bicoid. Now bicoid is responsible for determining the anterior posterior axis. But here we are talking about dorsal ventral polarity. So this also has no role. Now comes pipe. Now pipe, it is expressed in the ventral follicles and it is crucial for initiating the signaling cascade that leads to ventral fate determination. So pipe is again the correct answer. Why? Because it helps in ventral fate determination. So our correct answers are dorsal and pipe. I hope this question is clear to everybody. Okay? Is it clear to everybody? Okay, I hope it is clear. Now we'll come to the next question. Hmm. Just give me a minute. Hmm. So the question is, which of the following are the possible consequences of a loss of function mutation of kinase in motor activity? First, head alone will be absent. Second, it will fail to develop germ cells. Third, thoracic appendages will be absent. Fourth, irregular nanos localization. Fifth, it will fail to uptake maternal molecules from the nurse cells. And sixth, the last one is irregular bicoid localization. So this question will clear another concept. So try to answer this question. I will give you 2 minutes. Try to answer this question and then we'll try to see this. Hmm. Answer this question now. Two minutes to solve this question, okay? One more minute left. Okay. Shall I switch the screen now? Fine. Hmm. So let's see. Ah. So Ashika has answered F, Ajay has answered A and D, and Camelia has answered A. Okay. So let us see. The correct answers are hmm. The correct answer is it will fail to develop germ cells and irregular nanos localization. That means will fail to B and D. So the correct answers are B and D. So let us try to see. The question here says, which of the following are the possible consequences of a loss of function mutation of kinesin motor activity? So now kinesin, what is the function of kinesin motors? So basically kinesin is involved in transporting molecules along the microtubules and including those which are necessary for anterior to posterior axis specification. Okay, so remember that kinesin proteins are the motor proteins which are responsible for the transportation of molecules. So the molecules that have to go from the anterior to the posterior axis, so those molecules move with the help of these motor proteins. So therefore kinesin is very much involved in anterior to posterior axis specification. Now, what happens if there is a loss of function mutation of kinesin motor activity? So, if I say loss of function mutation, that means the function kinesin protein is mutated in such a way that it no longer can function to transport the proteins. Now, when kinesin is mutated, then what will happen? Anterior posterior axis formation would be kind of slowed down. So, but to say that only head alone would be absent, that can might not be true because its mutation does not specifically lead to the absence of head alone. It mutation can lead to even more some more circumstances. Although I understand that because in the posterior end, when your torpedo and crouple interact with each other, it leads to the formation of your motor proteins, and the negative end at the negative end, your bicoid kind of binds, and at the posterior end, some other proteins binds. That all is fine, but to vaguely say that if kinesin is absent, then only there will be a compromise in the head portion, that will be a wrong statement. Okay, it is not a very specific statement, so this is a false one. Then it will fail to develop germ cells. Will it fail to develop germ cells? Yes. Why? Because kinesin is very much important for the localization of germplasm components such as Oscar to the posterior side of the oocyte, and this Oscar is crucial for germ cell development. So, what it would be correct answer? Why? Because kinesin is responsible for the localization of Oscar. And this Oscar is involved in your germ cell development. Okay. Oscar is involved in your germ cell development. Therefore, if kinesin is not there, the movement of Oscar, the localization of Oscar to the posterior side would be compromised, and therefore it will fail to develop the germ cells. Then thoracic appendages will be absent. So thoracic appendages are specified by some other genes. For example, homeotic selector genes. And therefore, thoracic genes are less dependent upon the kinesin mediated transport. So, thoracic appendages are less dependent on kinesin mediated transport. So this is also not the correct answer. Then irregular nanos localization. Yes. Why? Because kinesin, it is involved in the transport of molecules such as nanos, which is localized in the posterior. So therefore it will when kinesin is not there, then nano's localization would be hampered, and when nano's localization would be hampered, that means posterior development would be hampered. So this is the correct answer. So C. So from here only we can say that A option is incorrect. Why? Because the loss of kinesin will inhibit nano's localization also, and nanos is responsible for a posterior development. That means not only the anterior portion is affected, your posterior portion is also affected. Then we come to the next option, that is it will fail to uptake maternal molecules from the nurse cell. Is this true or false? It is false. Why? Because the movement of maternal molecules from the nurse cell, it is not dependent on the kinesin motor activity. Okay? What I'm trying to say is, again, listen to my statement, that the movement of maternal molecules from the nurse cell, it is not dependent on kinesin motor activity. It is dependent upon cytoplasmic streaming and microtubule dynamics. So kinesin mutations specifically affect the localization of um, the zygotic determinants mostly, and they do not necessarily involve help I mean affect the overall uptake of maternal molecules. So this is also wrong. Then irregular bicoid localization. So bicoid is present at the anterior side, but its localization is dependent on dynein. Remember, bicoid localization is dependent on dynein and not kinesin. So, a loss of function mutation of kinesin will not hamper the bicoid localization as such. So, your correct options would be B and D. Fine. Okay, I understand it is a lot to take because in each there are no straightforward questions here. Like in every question, you have to apply some or the other kind of logic. But then, like this only, you will understand and you will be able to solve the questions. So there are many questions. We'll see how much we can cover because I don't want to, just for the sake of finishing off the questions, I don't want to hurry. Otherwise, you won't be able to understand. So even if we are left with one or two questions, there's no problem. We'll cover it in the next class. But I want you to understand the concept, and therefore I'm giving time on each and every question, because the main idea here is for you to understand the subject and not me to just hurry up and solve the questions, okay, and complete my work syllabus. Anyways, now we'll come to the next question, which is, which of the following regarding Drosophila development are true? First, loss of function mutation in torpedo adversely affects germ cell development. Second, effect of a loss of function mutation in the germline torpedo is seen one generation later. Third, Oscar mutants will fail to develop the germline. Then, bicoid mutation prevents germline development. Overexpression of pipe inhibits the dorsal fate. And last option is microtubule depolymerization will affect the anterior migration of the nucleus during oogenesis. So, 3 minutes to solve this question. Try to read each and every line carefully. Read, reread, and again reread. Then only you will be able to understand the meaning of the sentences. Okay? So I'm giving you 3 minutes. It's 7:32 right now. So after 3 minutes, I'll just switch the screen. At 35, I'll switch the screen. Okay? Now solve it. Read all the options very carefully. Hmm. You have 1 minute left. Read all the options. Okay, now I'm switching the screen. The time is up. Hmm. See what you have answered. So, okay. So Ajay has said E option. You didn't understood what E option says. Okay. And I don't know what. So you have opted C, F, and B. C, F, and B. E, you could not understand. That's what you're trying to say, right? Okay, so we'll see what are the correct answer. The first is effect of loss of function mutation in germline torpedo is seen one generation later. Oscar mutants will fail to develop the germline and microtubule depolymerization. So B, correct answer is B, C, and F. I think F is the correct answer, no? Yeah. So B, C, and F are the correct answers. C, F, and B. Yes, you have answered it correctly. E, what is E? So, okay, so now one by one, we'll try to understand each and every sentence. So, we have to just mark the true statements in this. So, the first, yeah. So, first is loss of function mutation in torpedo adversely, hmm, adversely affect the germ cell development. So, where torpedo is involved, where? Torpedo is involved in axis development. It is involved in axis specification. It is it is the mere function of torpedo, or the major function of torpedo is in axis specification. It is not directly involved in germ cell development. So this statement is not true. Then effect of a loss of function mutation in the germline torpedo is seen one generation later. True. This is correct statement. So torpedo signaling, it affects the next generation. Why? Because its role in oogenesis impacts the oocytes which are passed on the next generation. Okay, so always remember that torpedo signaling, it affects the next generation. So if there is any problem, if there is any mutation in the germline torpedo, its effect would be seen in the subsequent, I mean in the next generation, in one generation later. So this is the correct option. Okay, so remember this, that torpedo signaling, it affects the next generation because its role in oogenesis impacts the development of oocytes. Because the role of torpedo, it impacts the development of oocytes which are passed in the next generation. Fine. Then Oscar mutants will fail to develop the germline. It is true. Why? Because Oscar, it is very much important for the localization of germplasm and germline development. So without Oscar, the germline development is hampered. So this is the correct option. Then bicoid mutation prevents the germline development. No, see, bicoid is involved in anterior to posterior axis formation, just like your um, torpedo. So bicoid is involved in anterior to posterior axis formation, and they do not directly prevent the germline development. So that is dependent on other factors. So germline development can be affected by Oscar and to some extent nanos. So always remember that Oscar regulates germline development. So germline development is regulated by Oscar. Then next is bicoid mutation prevents germline development. No, this is also wrong. Then overexpression of pipe inhibits the dorsal fate. So pipe, where what is the function of pipe? So basically pipe is very much important for determining the ventral fate. So pipe protein is important for pipe is for ventral ventral axis specification. So pipe is involved in ventral expression. So if pipe is overexpressed, then there can be enhanced ventralization. But it doesn't mean the dorsal fate would be cut off unless and until pipe is expressed in the anterior, I mean in the dorsal side. So this is wrong because pipe, it is involved in ventral axis specification. So if pipe is overexpressed, there can be more ventral axis specification, it can be enhanced ventralization, but it doesn't mean that dorsal axis specification would be inhibited. Fine. Then next is microtubule depolymerization will affect the anterior migration of the nucleus during oogenesis. So, see, the, depolymerization means that the polymers are getting detached from each other. It is getting depolymerized. That means the individual polymers are getting detached from each other. So obviously microtubules are important for the proper migration of the oocyte nucleus. So if there is depolymerization, then depolymerization will prevent the migration, and this will ultimately lead to uh, 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 this will ultimately affect the establishment of anterior posterior axis. So this is also correct. So microtubule is responsible for proper migration. Okay, if it is depolymerized, if it is there is depolymerization, then what will happen? Proper migration is prevented, and it will ultimately affect the anterior to posterior axis formation. So this is it. So I hope this question is clear to everybody. Okay. Now we'll come to the next question. Hmm. E, about last question. Um, Last question E. It will fail to uptake maternal molecules from the nurse cell. What was the question? Kinesin. So see, we know that mostly um, uh, like the movement or the transport of molecules, they are dependent upon some kinds of microtubules and motor proteins. Okay, we know this. But when we talk about in the nurse cell, what are there in the nurse cell mostly? So in the nurse cell, mostly you have some kinds of maternal genes, and even if there are other genes which are present in the nerve cell, but the movement of molecules from the nerve cell is not dependent upon the motor proteins. Okay, I'm again repeating the statement. The movement of molecules from the nerve cell is not entirely dependent on motor proteins. Rather, it is dependent upon cytoplasmic streamings and some other kinds of, maybe to a certain extent, some kinds of microtubules, but mostly it is dependent on cytoplasmic streaming. Therefore, if there is a loss of function mutation in kinesin motor activity, the uh, maternal molecules movement from the nurse cell is not affected. Okay, so this is the explanation. For more detail, if you go and read the point, it has been given by sir also, or you can go in Gilbert's and you can check the point where they mention about how uh, gherkin and torpedo reacts with each other. At that portion only you can find a good explanation of this particular topic. When gherkin and when torpedo receptors, when gherkin comes and binds, and after that binding, your mostly your dynein and this kinesin uh, formation takes place, and in the negative end your bicoid comes and bind. But before that only some kind of movement has already taken place. Some kind of movement of the maternal molecules have already taken place. The maternal genes have already taken place. Then how is that taken place? Because microtubules, I mean the motor, these proteins formation has occurred at that time only. So before that, how it has taken place? Because of the cytoplasmic stream. So you need to go there and check. There we can find it in a better way. It is an oocyte. It is an oocyte. See, these nurse cells are present in the oocyte. Okay, so these nerve cells within the oocyte, these nerve cells are present, and these nerve cells have major role when the fertilization has not occurred. See here, if you see, I don't know whether it was there in this, or these are the lectures of your sir. Mm, no, it is not present here. Is it present here? No. Mm, yeah. See, this is the oocyte, and these are the nurse cells. Okay, and these nurse cells Whatever they secrete, the movement from these nerve cells is not dependent upon the motor proteins. Okay, here these are the torpedo, which are the receptors, and these green dots that you see, these are gherkin proteins. So when these gherkin proteins bind to the receptors, then your after the binding of gherkin protein to the receptors, the microtubule formation takes place, and then these are the microtubules that are formed, and these are polarized microtubules. On the on the negative side, bicoid mRNA binds. On the positive side, Oscar mRNA along with kinesin binds, and bicoid binds along with dynein, and Oscar binds with kinesin, and then this happens. So, but before that, these nurse cells, the components from the nurse cells, are moving without the help of the microtubule. I hope this is clear. You can just read, go back and read this part. It will be even more clear. Fine. Now we'll take the last question for the day. Okay, we have just two more questions left. I will take one more question, and other question I will take in other class. Okay, so I will take this question because I don't want to hurry up and okay. So this is a question. It itself is a very big question. So the question says, which of the following statements regarding Drosophila development are correct? So again, we have to select the correct statements. During oogenesis, the cell that has the highest number of contact points with the neighboring cells become the nurse mother cell. B. Blocking the ring canal formation prevents oocyte development. C. Germ cells are one of the first specified cells in the fly. D. Synthesis of maternal messages in the oocyte is important in access specification. E. Colchicine treatment within the oocyte will inhibit the anterior posterior axis but will not have any effect on dorsal ventral axis. And finally, last one, initial polarity of the cells is dependent on zygotic torpedo. 3 minutes, it's 7:50. At 7:53, I will switch the screen and then very quickly we'll solve this question and we'll end for the day. Now try to solve this question. Everybody, listen the options very carefully and then try to answer. Think. 2 more minutes left. One more minute left. Okay. Time's up. Hmm. So Ajay has answered B and C. This is the question. Blocking germ layer cells. Yes. So B and C are the correct answers. Now, quickly we'll try to answer, understand each and every options. So we have to select the statements which are true for Drosophila development. So first statement is, during oogenesis, the cell that has the highest number of contact points with neighboring cells will become nurse mother cells. No, there is nothing such as, because the cell that becomes the oocyte, it is determined by specific mechanisms. It is not simply dependent upon the contact points of neighboring cells. Okay, so this is a wrong statement. Then, blocking the ring canal formation prevents oocyte development. Yes, it is true. So these ring canals are nothing but they are important for transporting cytoplasmic material from the nurse cell to the developing oocytes. Okay, so which is crucial for oocyte growth and maturation. So this again proves the point that your motor proteins are not important for transportation of the materials from the nurse cell to the oocyte. So this is the correct statement. Blocking the ring canal will prevent oocyte development, because this ring canal is required for the transportation of cytoplasmic materials from the nerve cell to the oocytes. So if this ring canal is blocked, then all the cytoplasmic materials will not be transferred. And since all the cytoplasmic materials will not be transported, your oocyte development is prevented. So this is the correct option. Then next statement is, germ cells are one of the first specified cells in the body in the fly. Yes. Germ cells are among the first cells that are specified during earlier embryogenesis. Okay, this is also correct statement. Then synthesis of maternal messages in the oocyte is important in access specification. See, maternal messages, they do play an important role in access specification, but this is not the only thing which is responsible for access specification. Why? Because there are other regulatory processes that specify access beyond just the maternal mRNAs. So, although maternal messages are important, but just we cannot say, I mean, although maternal messages play a role in access specification, but they are not the only things that determine the access specification. There are many other factors that determine the access specification. So this is an incorrect statement. Then, colchicine administration within the oocyte will inhibit the anterior posterior axis, but will not have any effect on dorsal ventral axis. It will have effect on dorsal ventral axis only. Why? Because colchicine or colchicine, whatever you pronounce it, it basically interrupts the microtubules. So this colchicine, it interrupts the microtubule. So when microtubules are interrupted, that means both anterior posterior axis formation is also inhibited and dorsal ventral axis formation is also inhibited. Why? Because molecules, they need to travel from the anterior to the posterior end, from the posterior to the anterior end, from the dorsal to the ventral end, from the ventral to the dorsal. And the way through which they can move within the embryo is because of the motor proteins. Motor proteins are nothing like cars. So from one point to another point, if we want to go, when it is a large distance, we take a vehicle. Okay, we need to have a vehicle. So that vehicle is your motor proteins. And if that vehicle, we do not have a vehicle, we cannot move from one place to another just by walking if the distance is very far. So this colchicine is actually inhibiting or interrupting the vehicle, which in this case are our microtubules. So if the vehicle, that is your microtubules, are only interrupted, then your axis formation will not take place because molecules cannot move from one part of the embryo to the other. So this is also wrong because it will affect both anterior to posterior axis also and dorsal to ventral axis also. The last point is initial polarity of the cells is dependent on zygotic torpedo. See, initial polarity is primarily dependent upon the maternal factors, not by zygotic torpedo genes. So, whenever the term comes initial polarity, so initial polarity, the credit has to go to the maternal genes and not to the zygotic genes. So, this option is also incorrect. So, yeah. So, we'll end our class here. So I hope that the concepts, whatever concepts that we tried to discuss, you could understand the concepts. At least, even if you're able to understand 1% from the whole class, it's more than enough for me. So I would suggest all of you to please go and read as much as you can on this topic and try to solve more questions. You can get more questions from any MCQs available online or any CSIR questions that are available online, particularly on developmental biology. Then only you will be able to understand this concept, because it is a not a difficult concept but an intellectual concept. You have to apply your brain here. You cannot simply just uh, memorize the things. You need to understand and apply logic. Fine. So with this, we'll end today's class. We'll meet again in next week. And one question is left. It's a graphical question. So it will take at least 15 minutes. That's why I'm not solving it now. We'll solve this question plus the other questions in the next class. Okay. So bye everyone. Have a nice week.